Hello, everyone, and welcome to today's live broadcast, Finding Coherence in Chaos by Embracing Evolution and Complexity. It is presented by Kenneth Buetow, PhD, Director of the Computational Sciences and Informatics Program for Complex Adaptive Systems, and Professor in the School of Life Sciences at Arizona State University. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Buetow. I will now turn the presentation over to him.
Thank you very much. My name is Ken Buetow from Arizona State University. I want to begin by indicating that I have no disclosures relevant to this particular presentation, and move into essentially what's the fundamental research question that this presentation is attempting to ask and answer, and that's what drives common disease etiology and outcome. I'm a lifelong geneticist and genomicist, and this has been a question that has motivated my research career since the various early inception. We all know, and from our own personal experience, that different people with different circumstances respond differently to environmental exposures, and that risk for common disease is not uniformly distributed among the population. Shown on this slide, we can see a cartoonish example of that, where we know that some people, when they smoke, go on to have morbid outcomes such as heart disease and or lung cancer. But on the other hand, we also are aware that others seem to be able to have particular environmental exposures with impunity and have no ill health or morbid consequences. A brand new field in biomedicine has emerged to try to attempt to address this concept. And that's captured under the concept of personalized or precision medicine. As this audience is aware of, personalized or precision medicine is about delivering to the right individual the right care at the right time and the right place. And in personalized medicine, we recognize that the cancer community has been one of the fundamental pioneers in this space. Now, interestingly, given my historic background at the National Cancer Institute and as a lifelong cancer investigator, I would like to believe that this is actually attributable to our insightfulness in the cancer community. In fact, and arguably, nothing could be further from the truth. One of the reasons that the cancer community was an early adopter of personalized and precision medicine was the recognition that, unfortunately, many of our drugs, as shown at the bottom of this slide, just were completely ineffective. We knew that some individuals responded, but we didn't know when or why or who. So the cancer community was a very early adopter of the molecular definitions of medicine. And as a consequence, was one of the very early poster children. This is a magazine from almost 20 years ago now at this point, describing how we were using targeted therapeutics based on the molecular basis of disease to try to advance the field of cancer therapeutics and understanding of cancer susceptibility. Unfortunately, as an early adopter, being on the bleeding edge of personalized medicine, we actually discovered early on that, unfortunately, the simple approach of where we would find a mutation in a particular tumor and develop therapeutics to attack that particular mutation, as shown on this particular slide, while having benefit, and you can see nearly an immediate benefit in terms of survival. What we could see is at the end of the day, unfortunately, many of these therapeutics have been disappointing. And that in fact, the reason they're disappointing is that within weeks, in this instance, we can see the mean difference here is as few as 12 weeks difference, 12 to 14 weeks difference in overall survival with the treatment of this specific targeted therapeutic that addressed a particular single mutation in cancer. And as a consequence of that, the overall efficacy of personalized and precision medicine has been overall questionable in terms of changing the outlook of cancer. So why is that the case? Well, we actually know it's the case now with further characterization of cancer and its examination that cancer, when we look at it, is not a simple disease, but in fact is quite complicated, and I'll get to in a moment, quite complex. We now know that when we look at cancers across individuals, we can see that there are literally hundreds of alterations in cancer cells, and that these hundreds of alterations are working in combination with each other to produce the cancer phenotype. We've learned that these cancer mutational profiles are different among different individuals. So even though there's hundreds of individual cancer mutations, what we see is that very rarely do we see, and only in a small percentage, do these cancer mutations overlap with one another. More pragmatically, what we see is that within a cancer, the cancer itself is not homogeneous with respect to these hundreds of mutations, that in fact, the cancer tumor itself is a hodgepodge of many, many different cell types. As shown in the bottom right portion of this figure, we can see a given tumor actually is composed of essentially many, many different types of cells with many different types of mutations. And these different mutations then have the capacity to change over time and in response to the individual therapeutic intervention. So when we have these exquisitely targeted monotherapies, we do a great job of knocking out one type of these cells, but then very rapidly the remaining cells respond and actually compromise the outcome and health of the individual. This finding has actually generated some significant controversy in the community and some actually exasperation, probably best captured by one of the pioneers of molecular oncology, Robert Weinberg at the Whitehead Institute at MIT, who actually has, in some sense, thrown up his hands and suggests that cancer is just too complicated and that our current understanding renders cancer almost unapproachable. Well, I would actually argue, and again, as a student of complexity and as a member of the Complex Adaptive Systems group here at ASU, that actually this complexity is not unanticipated as what we now know from the molecular mechanisms, and in fact just requires us to look at the problem in a different way. And I'll use a metaphor of the pointillists, the flavor of impressionist painters at the turn of the 20th century. The pointillist shown here is a segment of a pointillist painting, and what you can see here is the collection of individual dots that actually underpin a given painting. And what we can see here is not unlike the circumstance we find ourselves in the cancer community right now. We now have the ability to characterize in exquisite detail the molecular characterization of every tumor, but what we see now is a disconnected collection of points, especially when we're looking at from this reductionist point of view, that our care is, what we care about is the individual points. But instead, if we actually pull back and begin to look at how the points interact with each other and how in combination and in context, the points produce a whole that's more than the sum of their parts, produce an emergent property, we can see that this, what appears to be chaotic pattern, chaotic randomness starts to emerge into a pattern. So if we pull back a little bit, we can see that these individual dots come back to actually form a particular shape and form. And if we look at how these combinations of dots in this particular figure interact with other figures, we can begin to see the overall emerging story of how this particular collection of dots or mechanisms or components then interact into a much larger story. And we can see that what was confusing and or uninterpretable at the individual dot level, when we look at the integration of these individual dots and how the collections of dots interplay with other sets of collections of dots, we can begin to understand the whole story. To do this then requires us to approach our phenotypes, our complex phenotypes, or our common phenotypes as complex systems. So let me take a minute or so to define what is a complex system. Well, complex system differs from a simple or reductionist view of the world by the fact that we care about the relationships, the interconnectedness, and the evolution of that system. The system itself learns and adapts in response to conditions. Now think back to what we saw with respect to this exquisitely targeted monotherapy, targeted therapy, and we could see that the cancer as a complex system adapted to this. But I think two key take-home lessons emerge from this. First, a complex system cannot be understood simply by taking apart and analyzing the individual components. Having or knowing the individual genes independent of their context does not help us or is not the answer to the problem. Knowing the individual components is an important first step, but we have to see how they work with respect to each other. And that's because what we see in complex systems is a phenomenon known as emergence, where combinations of things produce an outcome that's different than the individual effects. And those combinations, simply stated, 1 + 1 can equal 0 instead of 2, or 1 + 1 may equal 3. And because of that, what we see is that you can't simply fix a complex system by manipulating a single component. You have to alter its trajectory. And I think the best metaphor for this is shared on my favorite Google Doodle from that represents an Alexander Kaldor mobile. And those of you who have ever seen one of these mobiles in action, what you can see is touching any individual piece of this sets the entire sculpture in motion. And it's very difficult to predict how the action of any one specific leaf of this sculpture will result in its overall movement. And we might argue that in complex systems, we need to know both all of the pieces and how they're interconnected. So how does this change our thinking about looking at common disease and common complex traits? Well, first, if you're going to approach a complex adaptive system, one of the things we have to do is admit that this is in fact a complex system. This is surprisingly hard to do in the biomedical community that's been trained for now generations to use reductionist approaches, that the presumption that if we just find the gene for this or the gene for that, that we will have actually cured cancer or diabetes or obesity or multiple sclerosis, the idea that there's a single driving individual factor. So first and foremost, we have to change our global thinking. But it's not, once we do that, then there are some very specific steps as to how we can start to embrace complex adaptive systems and have an impact on these large phenomena in biomedicine. So approaching, first step is, the good news is that we're well along the way in this, in terms of our current paradigms, is that we have to inventory the components. We've now done a great job. We now can routinely get genomes and see how all those pieces and inventory all of the pieces and their individual states. But what we're not as good at yet is actually understanding how those pieces come together. And one of our critical components then in approaching complex systems is not only finding what the pieces are, but conducting the experiments that allow us to see how they interact with one another. Another novel feature, though, of complex systems is recognizing that it's dynamic. Now, when I say dynamic, that means, and as we saw from our cancer treatment example, is that it changes over time. It changes in response to its environment. It changes in response to intervention. So if we want to actually understand complex systems, we have to measure them not at a single point in time, but look at their trajectory, how they change over time. And more importantly, not only how they change over time, but how do they change with perturbation or with under different sets of environmental or therapeutic conditions. Lastly, and again, something that's been done for a long time in biomedicine, but we have lost somewhat, is we have to model the system. Now, I'm a geneticist by training and by choice, and what we recognize is that actually this is something that's been lost in the field of life sciences and biomedicine in general. While we're familiar with model systems like Drosophila or mouse model systems in biomedicine, we fail to recognize that actually much of our fundamental understanding in the life sciences has emerged from modeling systems. Arguably, Friar Mendel, as a geneticist, was one of the first modelers in the sense that he was looking for ratios when he was studying his pea plants, again, another model system of inheritance, to try to understand and identify what were the models of his time, ratios, 1 to 2 to 1 to 2 to 1, 9 to 3 to 3 to 1, all of these different Mendelian ratios that we all commiserate, that we all suffered with when we were taking our undergraduate genetics courses. So we've made progress in cancer. Shown here is a slide of where we stand with respect to cancer. Arguably, our reduction or leveling of the incidence cancer has been in spite of our efforts to develop therapeutics, as I showed on the earlier slides, largely probably attributable to changes in lifestyle, reduction in smoking, reduction in other key exposures that may perhaps had cancer increasing. So the general rule for cancer today, as we can see, is either flat incidence or slightly decreasing incidence, which is positive news. However, this is not the case for all cancers. And what we see is that, in fact, there are a number of cancers, a small number of cancers, that dramatically violate this premise. And I'm going to focus a little bit on one of these specific types of cancer, because this is what I'm going to drive the rest of my presentation around, our investigation of this other form of cancer, or one of the forms of cancer that isn't following this pattern. That would be hepatocellular carcinoma. So hepatocellular carcinoma is the 5th most common cause of cancer in incidence and the 2nd leading cause of cancer mortality. Hepatocellular carcinoma is the most common form of liver cancer, and in fact has a dismal 5-year survival, less than 18% 5-year survival. But more problematically is what we can see is over the last 2 decades, the incidence, last 2 to 3 decades, the incidence of hepatocellular carcinoma, primary liver cancer, has doubled in the United States. There's a number from the SEER registry in the United States, and we can see that in all races, white, black, Asian, Pacific Islanders, in each of these different groups, the incidence of liver cancer has increased. Interestingly, this is not just an American phenomenon, but in fact, a worldwide phenomenon. And in the except in the instance of Eastern Asia, where there have been efforts put to eliminating one of the primary risk factors of primary liver cancer, we can see worldwide, I apologize, difficult to read in this, but suffice it to say that any region of the world that you look at shows an increasing rate of liver cancer, similar to what we're seeing in the United States. So just to focus you with a red box, we can see here in North America the trend I showed you on the previous slide, but we can see even in places as distant as India, we can see that the rate of liver cancer in the last 2 to 3 decades has nearly doubled. So this is a cause for pause. Is liver cancer likely to be, for our generation and our children's generation, what lung cancer was to our generation and our parents' generation? As you all are probably aware, prior to the onset of the 20th century, lung cancer was a very rare disease. And it was only with the widespread distribution of smoking brought on by the world wars and other lifestyle changes driven by marketing and tobacco firms, we saw an explosion of lung cancer. Is this trend we're seeing in liver cancer going to be the same as what we saw in lung cancer? And if that's the case, what is it that's driving this change in liver cancer? Well, historically, liver cancer had 3 primary causes of its etiology: hepatitis C, hepatitis B, and alcohol. And you can see both of these are still tremendously high risk factors. And if one has a chronic infection for hepatitis C or hepatitis B, one has astronomically high rates of developing liver cancer. However, what's happened in the last 20 to 30 years is these risk factors have been supplanted worldwide with an additional collection of risk factors, that being obesity and type 2 diabetes. And what we can see now from information in the United States, and now also confirmed in the rest of the developed world and being explored elsewhere, is that these 2 exposures, obesity and type 2 diabetes, actually go on to have now the major attributable risk for developing liver cancer. Obesity and type 2 diabetes, oftentimes called diabesity, actually are the primary drivers of liver cancer rate, and arguably the major drivers of the increase in liver cancer. So why is it that obesity causes these? We know that actually obesity has multiple potential outcomes that include cancer, obesity, and cardiovascular disease. But what's unclear is, why do some people go down the path of type 2 diabetes, non-alcoholic steatohepatitis, a form of liver disease, and ultimately develop liver cancer, whereas other people can have an obesogenic phenotype with minimal morbid health outcomes, or others go down and develop cardiovascular disease, or others go down and develop different forms of cancer? What is it that's driving these different underlying paths of disease progression? Well, my group has been interested in using the role of inherited DNA variation to better understand what might be driving susceptibility to all of these risk-producing complex traits, common complex traits, and what may be driving the pathway down progression that we just saw from obesity ultimately to primary liver cancer. Now, the feasibility of this is actually well understood, in the sense that we know that phenotypes such as obesity and type 2 diabetes have huge underlying heritability. Arguably, they have a tremendous fraction of their total variation in the population that's driven by inherited genetic variation. Even liver cancer, as cancers go, has a very high heritability. And you can speculate, if you need these other precursor conditions, that you may actually even have, among those that have followed this path, even higher heritability. We've done countless genome-wide association studies to try to identify the basis of obesity, type 2 diabetes, but to date, there's been very mixed success in having reproducible findings. And arguably, this is because complex phenotypes are not the product of simple individual traits or individual genes. There is not a gene for obesity. There is not a gene for type 2 diabetes or a gene for liver cancer. But in fact, these complex phenotypes manifest as products of networks. Now, in cancer, we've known this for a while. We, again, dragged kicking and screaming perhaps into this point of view by looking and seeing that we couldn't explain cancer by looking at a single individual gene, but in fact, we had to look at combinations of genes to understand the complexity of cancer. So what my group has been doing is now using networks as our primary vehicle of analysis. And instead of looking for the role of an individual gene, we've been using networks as an analytic framework and embracing the emergence, the interaction between the individual components to then explore the susceptibility to the progressive phenotypes as well as the outcome phenotype of primary liver cancer. So we've done this by looking at a combination of publicly available genome-wide association data sets, 3 public data sets for obesity, 4 public data sets for type 2 diabetes, and a historic data set for hepatocellular carcinoma that we published on several years ago. Interestingly, what we have found is as we analyze these complex phenotypes, obesity, type 2 diabetes, and hepatocellular carcinoma, at a network level, recurring and simple patterns begin to emerge by embracing the complexity. Shown on this slide, I apologize for it being a little noisy, is a collection of pathways that we observe to be significantly associated with obesity, type 2 diabetes, and hepatocellular carcinoma. And what is striking about this is when we look within a particular phenotype, obesity being represented here by BMI, type 2 diabetes being represented by TD2, and HCC explaining liver cancer, what we can see is, unlike the standard genetic analysis, we're seeing recurrent collections of loci completely reproducible across data sets within a type, and then more provocatively, seeing that several of these pathways appear to underpin all 3 of these phenotypes. So we see, unlike the traditional genome-wide association study, high reproducibility for our significant effects by focusing on the biologic pathway as opposed to the individual underlying genes. But more provocatively, we then see, for instance, that there may be common underlying pathways that not only drive susceptibility to the end morbid types, but also that drive the progression through the morbid types. So let's focus for a second on what do these pathways look like. And one that perhaps is a little surprising for all of these types, for obesity, type 2 diabetes, and liver cancer, Axon Guidance. Now, I'll point out to you that pathways like genes, their names are not particularly descriptive of what they do. And I want to focus in a little bit. I realize this is a largely unreadable diagram, but you can look at it on your leisure afterwards, after the presentation. Shown on this diagram is one of the pathways that we use that's significant across all 3 of the, within and across all 3 phenotypes. And green indicates that they were genes that we were able to evaluate in our GWAS studies. Significant indicates that they were significant in the study, and red means that the same variants were significant across the individual studies. But let me blow this up a little bit. So let's focus in on a particular area. So let's focus in on this area. And what we can see is that each of these pathways is composed of modules. In this instance, you can see this module was the Wnt signaling pathway and particular components that were significant in that. We can also see that this pathway integrated another module shown here at the top. And this module then contained both the MAPK signaling pathway as well as regulation of actin cytoskeleton. So it becomes interesting to see these pathways. And if we go back to the other table, what one could see is that, in fact, Wnt and MAPK as individual pathways were also showing up. So these pathways are rediscovering common molecular signatures that seem to be driving all 3 of these phenotypes. What's interesting, given that observation, is that if we take the individual genes that constitute these pathways, interestingly, 97 of 99 of them create a master network, suggesting that there's a common underlying connection a biologic network that underpins susceptibility progressing from obesity to type 2 diabetes to liver cancer. Now, what is this network and how did it come about? Well, I might argue that if we want to better understand the origin of these networks and better understand how these phenotypes emerge in contemporary populations, that we need to embrace evolutionary thinking. This understanding of biology goes all the way back to one of the founders of modern evolutionary thinking, Theodoros Dobzhansky, who argued way back in the early 70s and others earlier that if we want to understand things in biology, we need to look at them in the context of evolution. So I would argue that the application of evolutionary medicine thinking gives us new perspectives for understanding the phenomena that we're observing in things such as type 2 diabetes, obesity progressing to type 2 diabetes to liver cancer, and also give us a handle to interpret the results and better understand why these systems might exist the way they do. So how might we introduce evolutionary thinking to this particular problem? Well, let's first start off is what has been the primary driving force of human evolution? Well, arguably, one could say that the thing that unites us as humans in our evolutionary development and adaptation has been the expansion of the human brain. And it's through the human brain that modern humans became modern humans. And shown here is just the dramatic increase in the size of the human brain during human evolution. This is quite striking when we look at the context of the brain size and neuron count. And even though there are organisms with quote unquote larger brains, an African bush elephant, for instance, has a much bigger total brain in terms of its total size. What we can see, though, is that the global number of neurons that are present in a human brain grossly outstrip any of the other mammals, and to be honest, any of our other primate cousins. So brain development has unequivocally been one of the major differentiators of humans in human evolution. So why is that important? Well, other than our adaptability and our capacity to conquer every niche of the planet, there's been a cost that's come with this. So having this big brain is expensive. Having a big brain requires us to feed that brain. And we actually know that the big brain actually is a huge energy pig. And that arguably one of the things that has facilitated the development of the human brain was the virtuous cycle, the positive feedback loop, that the more humans became successful at hunting and in group activity, the sizes of their brains were able to increase because they had access to more energy. But there's an interesting additional permutation that's a product of having a big brain. And that's that a big brain, at least a human-sized big brain, not only requires energy to feed it, but also requires a unique volume of lipids to construct it. So shown on this slide is just a cartoon that shows the overall surface area of individual type cells. So you'll see a typical human cell, although there's probably no such thing as a typical human cell, has a surface area of about 0.002 millimeters squared. However, if you look at the size of a neuron, or you look at the surface area of a neuron, you can see that a neuron has almost 100-fold greater surface area because of its dendrites that extend out. And then second off, what we can see is if you look at the most extreme components of these that underpin our larger nervous system, oligodendrites, this is not just 100-fold larger, but it's actually 1,000. It's 1,000 to 10,000-fold larger. So for us to build our big brains, we need both calories to produce it, but more perhaps pragmatically or provocatively, we also need special collections of lipids in order to build the brain. And there's been studies that have shown that if you are lipid deprived, that in fact, lipid deprivation can result in microcephaly and result in actually malformed brains. So we needed to actually, if we were going to build our big brains, we needed to build the cellular machinery, the basic biologic processes to support the development of that big brain. And in that, then, there has been an evolution of how long-chain fatty acids are produced in humans. And what we can see is even compared to our closest human relative, chimps, we can see that in an animal diet, compared to our great ape colleagues, our family members, we can see that our lipids are not burned as an energy source. And then in fact, when we consume long chain acids, they're made into longer chain acids, as opposed to being created or composed simply for the purposes of being used as energy consumption. That we actually have created specialized processes in our lipid metabolism that allow us to meet the needs of our developing big brains. Shown on this slide is actually specific mutations identified by the Ray et al. Group, where we could see specific alterations, mutations in the specific processing of omega-3 and omega-6 fatty acids have evolved and differentiate our lipid metabolism from great apes. And we can see that the lipid metabolism overall is fundamentally different in our great ape cousins than in our own human family. Why is that significant? Well, one of the key places that this lipid metabolism, as you can see, for instance, the elongation factors and others, where this metabolic processing is taking place is in the liver. And more pragmatically, we can see now, if we look at the liver now, we can see that this lipid metabolism is critically used in terms of producing different long-chain fatty acids, and is critically important in sugar and insulin management. So now we can see that the liver is central in maintaining fat supplies and regulating our insulin levels. So our question then was, did the evolutionary changes that were necessary for us to produce these long-chain fatty acids then result in us actually increasing our risk to liver cancer? Has our big brains then, the evolutionary alterations necessary to produce big brains and produce the lipids necessary, put us at risk for liver cancer? So to explore this, we went back to our pathway analysis, focused on the specific pathways that are associated with lipid metabolism, and in fact saw that in all of these cases that the lipid metabolism pathways all were significantly overrepresented in individuals who develop liver cancer as opposed to those who don't. Variation in these very pathways appears to be significantly different in those who develop liver cancer versus those who don't. We can further reinforce this finding by independent data by looking at gene expression. So in this instance, we can look at these same pathways by comparing their variation in gene expression, both from this using information from the GTEx consortium of human tissues, looking at liver samples from GTEx, and then comparing it to liver samples associated with the Cancer Genome Atlas, and look to see, is expression of these pathways different in individuals with normal livers versus individuals who have developed liver cancer. So comparison, I apologize for the small size here. We can see that if we look at these 5 pathways, we can see, number one, that they are significantly different in normal liver from two individuals who go on to develop liver cancer. And interestingly, what it suggests is that these are predisposition pathways, that in fact, it's not a product of the cancer that's changing these, but the individuals who have liver cancer, go on to develop liver cancer, have fundamentally different gene transcription profiles in several of these pathways. And then in fact, the logic of these pathways, the lower panel of this slide, is different in individuals who go on to develop liver cancer from normal individuals in the general population. So this suggests that germline variation that we saw in terms of variation that could be due to evolutionary forces to develop big brains are resulting in different lipid metabolism in individuals who go on to develop liver cancer from those who don't. So shown here is just focusing on the specific genes, and we can see when we look at the liver cancer susceptibility pathways, to remind you of the ones we saw that are different from our evolutionary cousins, the great apes, we can see that in this instance, these elongation factors and other key variables associated with lipid metabolism are critically different in liver cancer as opposed to individuals who are not at risk at delivering liver cancer. So this suggests an interesting possibility. If we look at these particular pathways and assemble them as we had done with respect to our other investigation of the overall liver cancer susceptibility, what we can see is that in fact, these pathways again form a master network, and the red dot that's unreadable, I apologize, on this slide, actually brings in an interesting gene that may be of significance. That gene is the 3-hydroxy-3-methylglutarate coenzyme reductase gene. And that gene is the target used for treating cardiovascular disease by statins. So perhaps this suggests that we could have effects in liver cancer by using statin treatments. So again, in this instance, HGM coreductase is the primary target of statins that are used to reduce cholesterol, to manipulate cholesterol in individuals with high cholesterol. Interestingly, though, this gene is at the hub of a variety of critically important processes. So it not only manipulates your lipid levels in your liver, but it's been observed that manipulation of this gene has anti-proliferative, pro-apoptotic, anti-invasive, and cytostatic effects, and could be a master regulator of many different underlying processes. So this suggests that statin treatment could be an effective way to actually intervene in liver cancer susceptibility. So to explore that, a number of epidemiologic studies have been conducted. And by looking at individuals who were taking statin use for other purposes and monitor them in such things such as the UK's Clinical Practice Research Data Link, in a paper published in 2015, we can see that in fact, individuals who are taking statin do have a reduced risk of overall developing liver cancer. Now again, there's many confounding possibilities as to why this could be the case. I'm happy to answer questions related to this, but this is quite suggestive. The more definitive case, though, may be made by examining clinical trials or a specific clinical trial where individuals with liver cancer have been treated with statins after resection and seeing their recurrence rate. And what we can see here is from this particular Japanese study that when statins were administered to individuals shortly after their HCC surgery, that the risk of recurrence was reduced by half. So again, striking but still preliminary results in a relatively small study. So with that, I'd like to summarize saying that we think that network analysis finds common biologic processes within complex traits, that this systems approach allows us to have insights that the reductionist individual gene approaches don't develop. these network analyses identify common processes along disease progression paths. So we can not only find the underlying etiologic basis, molecular basis of these traits, but perhaps indicate how things might move forward or who is likely to progress down increasingly severe morbidity and ultimately have mortality-causing outcomes. We can see that the biologic processes are both expected, energy production, and to be honest with you, somewhat surprising, although in an evolutionary context, maybe we're not as surprised that Axon Guidance might be one of our underlying processes that's driving susceptibility from obesity to type 2 diabetes to liver cancer, and that there may have been evolutionary trade-offs associated with the development of the large human brain. And then lastly, that evolutionary thinking provides novel hypotheses to test and suggest risk-reducing interventions. So with that, I want to thank the people who have actually facilitated doing these studies, my ASU colleagues, Yikong Liu, Chantal Farrell, Madeline Stark, Jacob Brill, my former colleagues at NCI, and then the individuals who have donated and participated in the generation of the original Korean case control study. And close then with happy to take questions. And as always, our group is interested in collaborators and partners who might be interested in exploring any or all of these individual phenomena. So thank you very much.
Thank you, Dr. Buetow, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Okay, the first question is, why do networks of SNPs show consistency across data sets where individual SNPs don't?
Thank you very much. That's an interesting question, and I think one that maybe underpins the whole root of this talk, and that's the recognition that genes work in partnership with one another, and that the pattern of variation we see with genes, while individual variants produce risk, it's the combination, the interaction, the higher order terms of these interactions that seem to be what is crucial in producing risk, that it's combinations of the SNPs that are working together, and that these individual variants, when observed in isolation, can vary from individual to individual and from case and or control, but only when we look at the combinations of them do we have individuals who have everything that's both necessary and sufficient to develop cancer. So they don't mitigate or cancel the noise if we look at them in combination.
Thank you. What does the gene expression pathway analysis tell us that the SNP analysis doesn't?
So one of the advantages that we have for the gene expression analysis is the gene expression analysis is complementary to the DNA variation. So what we're seeing actually with the expression analysis is what's actually happening in the tumors, by the tumors and in normal liver. So the gene expression analysis tells us what the normal patterns would be within these particular processes, and then allows us to contrast whether the transcriptional differences reinforce and support the information driven by the DNA. So you can argue that the transcriptional information both is an independent validation of what we saw for the DNA variation, but it's also then represents an additional insight because it's done at a different level of analysis than what one sees with respect to the DNA-based variation.
Thank you for that. Your next one is, what do the pathways tell us about mechanisms of progression?
So thank you for that question. It's actually, I think the pathways give us novel biologic insight as to what actually is driving the specific susceptibility to the particular traits, but then clues as to why these traits may be connected in progression. So for instance, we can see that lipid metabolism certainly would make sense as a driver of obese phenotypes, but the connection of lipid metabolism to energy metabolism, in particular insulin, the regulation of insulin, as well as what we could see when we looked at, for instance, Axon Guidance and its connection to cell proliferation, as well as connection to immune response, may then be critical drivers that having alterations in one component then makes one susceptible to multiple different complex outcomes.
Thank you. Looks like we have time for one more question, and it is, could this approach be applied in other common complex diseases?
Yeah, thank you. I think that's an interesting and provocative way to take this work forward, is the observation that, in fact, these pathway-driven approaches actually can give us insight, not just in this particularly narrow area that we've looked at today as proof of concept, but in a variety of other complex traits. Could be useful in looking at phenotypes where we've found literally, again, dozens to hundreds of non-reproducible single nucleotide polymorphisms, SNPs, that are difficult to understand what their patterns. By integrating across these networks, we may get clues into neurodegenerative diseases like Alzheimer's disease or multiple sclerosis or other autoimmune diseases or cardiovascular disease. So I think that this approach to looking at the integrated phenotypes has been barely examined to date, and I think could be very profitable in examining a variety of different other common traits that are huge public health challenges.
Well, I would like to once again thank Dr. Buetow for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 10th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon.